Epigenetic reprogramming, particularly through the use of Yamanaka factors, is rapidly emerging as one of the most exciting frontiers in anti-aging science. At its core, this technique aims to reset the biological age of cells, not by replacing them or merely slowing down their decay, but by reversing many of the molecular signatures of aging itself. By partially reactivating the genetic programs associated with pluripotency, scientists have shown in recent animal models that it is possible to restore youthful epigenetic profiles, improve tissue function, and even extend telomere length, all without inducing the loss of cell identity or causing uncontrolled cell division. Yamanaka factors, October 4th, SOX2, KLF4, and CMYC, are the four transcription factors originally used to convert adult cells into induced pluripotent stem cells, iPSCs. While full reprogramming erases cell identity entirely, recent research has focused on partial reprogramming, transient, controlled exposure to these factors that reverses key hallmarks of aging while preserving cellular function. This delicate balance appears to rejuvenate cells at the epigenetic level, including restoration of histone modifications, DNA methylation patterns, and improved mitochondrial function, all without pushing the cells into a pluripotent or cancer-prone state. Animal studies have delivered compelling proof-of-concept results. In a widely cited 2020 Nature study, researchers demonstrated that cyclic partial reprogramming in aged mice not only improved regenerative capacity in muscle and pancreas tissues, but also extended the lifespan of progeroid, prematurely aging mice. Telomere lengthening was also observed, suggesting that partial reprogramming may reactivate endogenous telomerase or otherwise protect telomeric DNA through better epigenetic maintenance and stress resilience. These findings hint at a coordinated rejuvenation effect that goes beyond superficial molecular repair. It appears to reset aging at multiple, interdependent biological levels. Beyond just telomeres, partial reprogramming addresses one of the central causes of cellular aging, the accumulation of epigenetic noise. Over time, cells lose their tightly regulated gene expression profiles due to age-related disruptions in DNA methylation and chromatin architecture. This leads to a breakdown in cellular identity, increased inflammation, impaired repair mechanisms, and vulnerability to disease. By reestablishing a youthful epigenetic configuration, partial reprogramming may help cells remember how to function optimally, reviving tissue integrity, enhancing repair, and reducing markers of senescence. Importantly, this approach appears to influence many aging pathways simultaneously. Unlike targeted drugs or supplements, which may address oxidative stress or inflammation in isolation, partial reprogramming seems to trigger a broad systemic reset. Improvements have been documented in mitochondrial dynamics, proteostasis, protein folding quality control, and immune cell function. This is consistent with a more global resynchronization of gene networks, which may be why even short exposure to Yamanaka factors produces disproportionately large biological benefits in early animal models. Despite the remarkable promise, this field is still very much in its experimental phase. Achieving just the right degree of reprogramming is a delicate task. Too little may be ineffective, too much may risk erasing cell identity or triggering malignancy. The use of CMYC, a known oncogene, also raises safety concerns in therapeutic applications. Researchers are exploring alternative reprogramming cocktails, such as omitting CMYC or substituting it with less risky factors, to improve safety without losing efficacy. Delivery is another challenge. Systemic delivery of reprogramming factors in vivo requires precise control, both in dosage and in timing. Gene therapy vectors such as AAV, adeno-associated viruses, are commonly used, but ensuring tissue-specific expression and preventing immune reactions will be essential for translating this into human therapies. Some researchers are also investigating small molecules that can mimic the effects of transcription factors, which could pave the way for drug-based rejuvenation without the need for genetic manipulation. The ethical implications of epigenetic rejuvenation are also significant. If this technology proves safe and effective, it could redefine our approach to aging, shifting the paradigm from disease management to true biological regeneration. Questions about access, longevity limits, and intergenerational equity will inevitably follow. Still, the appeal of a non-invasive, system-wide method for reversing cellular aging is undeniable, and the early science is increasingly hard to ignore. In summary, epigenetic reprogramming through partial expression of Yamanaka factors represents a breakthrough concept in longevity research. By restoring youthful epigenetic patterns and reactivating protective genes, potentially including those responsible for telomere maintenance, it offers a way to turn back the molecular clock without replacing tissues or risking complete dedifferentiation. While technical and safety challenges remain, the trajectory of this field suggests that partial reprogramming could one day become a cornerstone of regenerative therapies, offering not just longer life, but restored vitality at the cellular level.